Hello students, welcome back to Catman Academy, and today we are going to discuss about plasmid again. And in the plasmid topic, today we are going to discuss about its important parameter, that is size of the plasmid and copy number of the plasmid. Right? Why we need to discuss size and copy number of the plasmids? Because these are very important in case of the gene cloning, which is done in variety of labs all over the world, right? So if we talk about the size of the plasmid, so it ranges from, I'm writing here, the size of the plasmid, it ranges from 1 kb to 250 kilobase pair, right? And from all these plasmids, only few are responsible, or you can say the few are useful for the cloning purposes under the circumstances, special circumstances, right? So for the making of a better cloning vector, we need a cloning vector. Its size should be less than 10 kilobase pair, right? If we talk about the copy number, in case of copy number, it depends on number of molecules of the plasmids which are present in the host bacterial cell. Host bacterial cell, right? So in case of the larger plasmids which are present in the host cell, they actually have less number of the plasmids. Larger plasmids in the host bacterial cell. Host bacterial cell, they have lesser number of the plasmid molecules, right? And in case of the smaller one, plasmids, so the bacterial cell which contains smaller plasmids in them, they actually have more copies of the plasmids, or you can say higher copy number of the plasmids. It could be 50 or it could be more than 50 plasmid cells, plasmid DNA molecules per cell, right? And in case of the cloning purposes, only these type of plasmids are useful because they have higher number of the copy number, and these type of plasmids can be exploited in the recombinant DNA molecules production. Right? These type of plasmids which contain high copy number, they are also called relaxed plasmid. Relaxed plasmids, right? So I talked about the size of the plasmids, and it ranges from 1 kb to 250 kilobase pair. So let's talk about some of the plasmids and their sizes. So for example, PUC8 plasmid, it has a nucleotide length in kb which is 2.1, and the host organism in which it is present, that is E. coli. Right? If we talk about another plasmid that is called E1, it has a nucleotide length that is 6.4, and it is also present in the E. coli organisms. Then we already saw RP4 plasmid. So it has a nucleotide length of 54 kilobase pair, and it is present in the Pseudomonas and other organisms. Right? Then we have F type of plasmid. It has nucleotide length of 95.0 kilobase pair, and it is present in the E. coli. Then same, we have tall plasmid. It has a nucleotide length of 117 nucleotide base pair, right? And it is present in the Pseudomonas. Then we have another one which is PTI ACH5 plasmid, and it has a nucleotide length of 213 kilobase pair, and it is present in the Agrobacterium tumefaciens. So this is the range of the plasmids which is present in different organisms, and it ranges from 1 kilobase pair to 250 kilobase pair. And some of these plasmids are actually very important in the gene cloning purposes, right? So for today, that's all we have. Thank you.